really, really exciting to be here and hear all the amazing applications of long read technology and Revio and fusion transcripts and disease and rare childhood disease detection. It's really just amazing as a father of young children. Um, I'm so excited to hear those things. And where we sit, and it's really pretty powerful that PacBio extends the invite to me to talk about what we really care about, which is how do we nourish our bodies? So our temples, those things that we care so much, how do we actually nourish those things? And up here on this slide, I'm just showing the really talented group that I get the privilege to work with at Hudson Alpha. And we call ourselves the Genetic Innovation Lab. That was a name that the individuals in the lab came up with themselves because I didn't really want to call it the Clevenger Lab. I didn't really like that name. So this is a better name. And what we do, we're really passionate about how do we democratize genomics for all of the crops and animals that people care about. And one of the things that really strikes me, I get the privilege to get to travel the world pretty extensively. And every time I go a new place in a new continent, I'm always looking at what crops are grown, how they're grown, where they're grown, and how much they're grown. And if you think about it, every single place in the world, there are human beings that care about how well those individual plants that they care about in their communities are growing because they either feed their families, feed their communities, or provide the money that they use to feed and take care of their families. And recently, 2 weeks ago, I was in Malawi. And what struck me was pretty striking is that every single inch of space between any tree, between any building, between any home, they were growing crops, and mostly corn at that point. It was harvest season. And all of those crops are harvested completely by hand. So children and women were out in the 90-degree weather and the beating sun, shucking corn off these corn stalks and harvesting tobacco, which apparently I didn't know, but it burns your skin because of the nicotine. And so if you think about it, the resources that corn has to provide these amazing sort of corn stalks that if we provide all the inputs for and we put them at the right planting density and we have the GPS controlled tractors, we can harvest them at this great efficiency. Huge swaths of the world don't have access to that technology. So even for that crop, you need to be able to develop better varieties for all these different places in the world. And so that is just a large commodity, but there are so many different plant and animal species that people care about that support the lives of humans across the globe that don't have those types of resources. So what we try and do is provide those resources for those groups that don't have large consortia, that aren't backed by global companies, that aren't backed by large NGOs that want to fund these types of research. And so if we take a step back and we look at in plant biology and plant improvement, animal improvement, how genotyping, how genomics are used in those spaces, historically and still now, they're using these types of technology. So either a fixed array, so in animal breeding specifically, they're doing millions of samples of fixed arrays in plant breeding as well. So those technologies have the limited application that you can develop them once. And then if you try and use them in new populations, if you heard about family-specific genomes, right, and how valuable and how important they are, the variation doesn't make as much sense. There's other applications of using sort of limited marker sets or reduced representation marker sets with RADseq or GBS or capture-based technologies to the tune of millions and tens of millions of samples a year. But again, you have limited ability to do any discovery or any sort of have the resolution that you need to make the real improvement. And on top of that, if you are a group that cares about Bambara groundnut, which is only grown in one country in East Africa, you need to have 50,000 samples that you're going to run to get a price that's affordable for you. So here's an example of how this works. So the Gates Foundation funds these excellence in breeding technologies, right, which are marker sets that are subsidized for breeders in different countries. What happens is that they have a set of markers that they can get on their populations for $2 a sample. However, in the vast majority of populations, and these things aren't said out loud, they're said sort of when no one's around, is that in the vast majority of populations, those marker sets are monomorphic, right? So they're completely unusable. So sort of like this idea of you should use marker technology to accelerate the breeding of your plant species doesn't make a lot of sense in all of these situations. So with that being said, when I started at Hudson Alpha, I came from a global food company, Mars Wrigley. They make Snickers and M&Ms. I wanted to really create a platform so that we could use genomics across all of these different plants and animal species. And as a breeder, as a breeding PhD, I knew that I didn't want to use arrays because I developed a lot of arrays and I didn't want to use them anymore. I didn't want to use GBS or RADseq or capture-based method. I wanted to use whole genome sequencing. So the whole platform is set up to, at scale, utilize low-pass whole genome sequencing. At first, we're using short reads, right? Because at that time, that's before the Revio came out. And so we do a type of genetic analysis that when it's right, and this is an example in watermelon, this is a new, there's a new disease in watermelons, a virus that is vectored by aphids. And when you do that analysis, it looks like Khufu pyramid. So that's why we named it that. And then in the last 2 years, we've had the distinct pleasure of working on over 65 species to the tune of now nearing more than 50,000 samples, mapping traits, identifying genes that are targeted to those traits, and then utilizing them for selection and breeding programs across plants and animals. So we're really sort of putting the money where our mouth is to help sort of provide these resources to groups all across the world. And here's a perfect example of that. So I love peanuts. I work on peanuts in my, I guess, day job, or not day job, my side gig, if you will. Peanuts are amazing because they're full of nutrition and vitamins and minerals and healthy fats, and they're cheap. They're cost effective. So in places of the world where you cannot afford pecans, it's not like pecans better. I'd rather eat pecans than peanuts, I must say. But when you can't afford those nuts, those resources, you can still buy peanuts, and most of the world can afford peanuts. And actually, it's one of the top 12 commodity crops in the world, and top 3 in oilseeds. So here's a disease in Argentina. So it's peanut smut disease. So when I was working at Mars, they started having problems with peanut smut disease. So if you think about global supply chains and you want to sell peanut M&Ms to every country in the entire world, which is kind of wild, but every country I've been, they've had peanut M&Ms in that country. And it's pretty weird. Even when I was in Malawi, I could buy peanut M&Ms. Thought that was pretty strange. So you have this disease. The spores are incredibly resilient and mobile. They're going to stick to your clothes. They're going to stick to your equipment. If you move that disease around the world, you will devastate the production of this crop, right? You're devastated everywhere. Every single peanut variety that is grown across the world is susceptible to this because it just pops up. And kind of like in a strange twist of fate, you can actually grow peanuts kind of everywhere. So a lot of the varieties that are grown are shared. Like in Australia, they grow varieties that were bred in Florida. In Africa, they have a lot of varieties that are bred in Texas. And so there's a lot of shared germplasm there, and it's all susceptible, okay? You can't move those spores to the United States to breed for resistance, to test for resistance, because if you did that, you're introducing them now into the place you don't want them to be. So how would you try and develop resistant cultivars or develop resistant varieties for this problem? You have to do it on site, and then you have to be able to quickly and effectively identify that functional variation, that variation that's attached, that's tightly linked to that trait, right? Disease causing, or resistance causing in this case, in the site, and then be able to breed for it with markers. So there's the actual field where we had 1,000 F2 plants. So in the plant breeding world, an F2 is just the families that come from the hybrid. So we make a hybrid, which is an F1. We self that, and those are each distinct families that we can do genetic mapping in. And we were able to utilize the technology we developed to sequence all of those F2s and quickly identify the gene that controlled that resistance. Now, we were lucky. It was a very strong major gene that controlled the resistance. And so when we go back and look at that variation in a set of diversity, which is on the left, we can see that when we have one allele in smut infested fields for 4 years, it's 0% infestation, right? So this is a gene that has major effect on this trait. And when you don't have when you have the opposite allele, it's horribly susceptible, smut all over the place. And because we can harness these technology, right? We're not limited by the information that we have. We can take that same population that we discovered the gene in, and we can make relevant breeding decisions. So on the right is the distribution of all the lines that have the resistance. And then how much of the background is the elite genetic background. So if we talk about things in crops that are important, being able to grow at a higher density, being able to have the higher quality, sensory, right? Some of you eat peanuts. You might have eaten peanuts that you didn't think tasted so good. You might have eaten peanuts that you thought tasted pretty good. All those things are controlled by numerous quantitative factors. So if we want to breed varieties quickly that we want to grow and want to harvest and produce products with, we also have to keep that in mind. So this is really powerful. So this particular breeder in Argentina, and then as well as 5 different breeding programs in the United States, which is funded by Mars and the USDA. We are actively now selecting for smut resistance and accelerating the process by saying, these are the lines you want to backcross again. And in 2 or 3 generations, you now have a disease-resistant variety that has all the characteristics that you want. So we're here. It's PacBio. 2 years ago, a colleague of mine, Justin Vaughn, we're talking, and he was like, man, it'd be super cool to do the long read low pass, to do the low pass sequencing with long reads. And I was like, yeah, but never going to be able to do that. And then you guys dropped this. And now we're like, wow, now we can actually think about doing that. So what are the things that normalizes that? We're going to hear more about that today, and we've heard incredible applications of that earlier. But one of the things is that we normalize pangenomes, right? So as a geneticist and a breeder, the application of pangenomes are incredibly exciting. And we have examples of this every day. We're working with folks, the kind of insights that you can gain. And I had nothing to do with the human pangenome reference, of course, but I'm just showing that to show you that we took, at Hudson Alpha, we have neurodegenerative disease researchers. So we have a large cohort of South American individuals that they've sequenced on arrays and full genomes. When we took those genomes and subsampled them down to 1x coverage and then ran them on the pangenome graph, the phase pangenome graph, the imputation is almost perfect, right? So at sites where it's like up to 90% missing data, you're almost perfectly imputing all those variants because your pangenome reference actually builds in that reference graph mentality. And then if we look at how concordant are those with the array calls, our quote unquote truth set, which is someone that works with arrays. I don't think they're always correct, but that's a nice truth set we can have. It's almost 100% concordant. So the point is that utilizing these technologies, even with the short read skim-seq, which I don't want to use, but in applications where still you need to do hundreds of thousands of samples on the research side, you can provide really accurate results on phase pangenome graphs, incorporating all that structural variation in the SNP data, right? So really powerful insights that you can be had. But I don't do human research. I work on plants. And so I'm going to tell you a story of how long reads are sort of elevating this little corner of the world and the sorts of things that we can think about moving forward, how we do plant and animal improvement for the world. So we have, and actually on Monday, we just broke ground on a new 45,000 square foot facility in Dothan, Alabama. Dothan, Alabama is in the Wiregrass region. We call it Hudson Alpha Wiregrass. That particular region is the home of George Washington Carver. Tuskegee is about 40 miles north. So it's a special place in my heart. One of my heroes of all time, George Washington Carver. He was famous for promoting peanuts in that area. In fact, there's a town, Enterprise, Alabama, where they used to grow cotton, and the boll weevil decimated that area. And the farmers there were some of the first to pick up peanuts at the behest of George Washington Carver. And they were so successful that they put up a statue in town to the boll weevil. Thank you, pest, for showing us the way. to grow peanuts. So I think that's really cool. So in this area, what we do is we incorporate our breeding program into the high schools, because 50% of the peanuts that are grown in this region, 50% of the peanuts that are grown in the United States are grown within 50 miles of Dothan, Alabama, right? So in Georgia, Alabama, and Florida. So almost everybody in their communities rely on peanuts for some reason. They're processing, they're trucking, they're growing, they're shelling. Peanuts are everywhere. If you have a peanut allergy, you don't want to live there. There's peanut dust all over the air, right? So these kids think that agricultural genomics and agriculture jobs in general, you just become a farmer. There's nothing else to do. So they don't know what sort of things they can do as scientists. So we brought the breeding program to them to show them how molecular biology and how genomics impact agriculture and sort of things that they might think about doing in their lives. So each child gets a peanut seed that they grow. They take data on. We go to the classroom after those plants are grown. And by the way, they're grown in the classroom, so they get to see them every day and take care of them. They name them fun names. We go in, we teach them how to extract DNA. So what that means biochemically, the science behind it, and then we actually do it with them. That's a really rough week. We're in classrooms about 8 hours a day for the entire week. We're right now in 12 high schools. So every semester, it's 150 to 200 students. Then we sequence that DNA, we analyze it with the students, and they become the breeders. We talk to them about what sort of things they might want to select for, and they make the selections as a class. And I have to adhere to that because I promised them I would. So it's a really sort of engaged program in those high schools. And really, like, what happens is I talk to the kids about what we're doing and how impactful it can be, and how many people actually worked hard to produce genomes, and companies like PacBio that produces the technology that allowed us to sequence the peanut genome and do all of these things, the history behind it and the impact that they can make. And they get so serious and they have so much fun, and they really, really enjoy learning about how molecular biology impacts their communities and also the globe. And so there's always a lot of smiles. And on that sign, you can say, we are using DNA to make better peanuts for the farmers of the Wiregrass. I'm going to tell you also that they're also doing that to make better peanuts for the world. And I'll show you how that works. So why am I talking about this when we're talking about long read low pass? Well, that's because of the ability to have that incredible throughput of really high quality hi-fi reads on the Revio, we are now doing the entire breeding program with long read sequencing. So not only are they sequencing all those and learning about it and using them for selection, but they're doing it with this latest technology. And it's just really just this semester was the first time that we did it. I'm really, really excited about that. So what did they do with the data? This is an example of what they might look like. These are just different lines that they grew. And then we're able to actually select for all these different disease resistances. So not going into the population and how we put it together, we can select for multiple packages of diseases. Because we sequence all of these lines and we get seed from them all, we can always go back and reanalyze when we learn new things. So I have projects looking at low aflatoxin and drought tolerance and different diseases. We can go back and now make new selections, okay, because we have all those data. And that's really the power of having the whole genome data on all of those lines. So in here, we actually have lines that have multiple disease resistances. They have the background of the elite parent, really high percentage, just incredible material. And after just 3 semesters, we have our first yield trial of a plant that has high oleic acid. It has resistances to 4 different diseases, including smut, which isn't even on the United States radar yet, but it will be soon. And it's just an incredible thing to be able to do, because if we take a step back, and I know many of you probably haven't thought about how breeding is done. If you take a step back, you have a limited number of seeds to test. So if I make a cross and I want to understand what are the best lines to make the selections of, I can only really test for yield and grade and quality and maybe one disease, because if I try and test for many diseases, all of a sudden I've lost all those seeds, right? Because I have limited number of resources available. So you really can't make selections for multiple diseases at once. You can only do that with marker technology, and you can really only do that when you have the ability to have all of that information at your fingertips. Looks like it. Oh, wrong thing. All right. So how can we do the long read, high throughput long read sequencing? Well, there's two actual ways to do it. If some of you are familiar with Seqwell, they have this really great technology that they just released recently called Longplex. So we use Longplex in some applications. They use their transposase technology to shear and then add a barcode at the same step, and then you can make the one microbelt prep out of however many samples you want to multiplex together. In Peanuts, we can do 12 for a Revio run. The size of the inserts is still not perfect yet. It's around 8 kb average, but they're working on moving that up. So we're excited about that getting higher. But all of the really great work that PacBio has done to do high throughput shearing and prep, we use the Hamilton Microlab prep, and we can do 96 well plates at a time. It takes about 2 hours, and it does mechanical shearing, and it is so precise and even. It's an incredible thing that you can do. That is a large sort of outlay of cash in the beginning for different folks, but that's what we're able to do. And then using the HiFiPlex library prep technology, which is really easy, then we can start multiplexing those samples. So that has given us the ability to actually do this in real time in a breeding program. So this is a really good example. So this is how I'm going to show you sort of how we can do things in one space and share them globally. This is the resistance to tomato spotted wilt virus, and it's bad in the southeast of the United States for peanuts. Researchers at the USDA and University of Georgia and others have tried to map this resistance for 20 years, right? They use SSRs, they use indels and rapids and AFLPs on the same populations with the same strong source of resistance. And if any of you are familiar with plant breeding at all, you know that anytime there's strong resistance, it's always in a background that is dog poo-poo, right? Like it's never going to yield well, probably tastes crappy, like wild species of tomatoes and things like that. So there has to be a ton of breeding and selection to take resistance and put it in a good variety. When we were able to create a pangenome graph 2 years ago using HiFi sequencing and then map onto that pangenome graph, we tagged this insertion immediately. And this insertion is the causative variation for resistance to this virus. And so utilization of those data allowed us to immediately pick it up. And then using long read sequencing in the selection allows us to tag it immediately as well. So now we can do things like, I'm just showing you here. So some people want to know what is the difference between sort of low pass with short reads and low pass with long reads. Well, there's a huge difference, of course. This is all mapping on a pangenome graph. So what's really fun is actually taking the long reads and genotyping them on an actual pangenome graph and seeing all the amazing insights that you get from that. So just if I just took the samples that I would have sequenced normally and the depth I would have sequenced normally, on the purple side, the purple color is what our yield is based on the potential variance in the graph. The black is the short read sequencing that we did on this program, just to show a comparison. And then the pink is actually when we subsampled down to the same depth. So doubling sort of your yield, doubling your resolution and your insights using the technology. Okay. So going back to that virus resistance. So I'm part of a group. It's called the Groundnut Improvement Network for Africa. And it is a group of 15 national programs supported by USAID that share germplasm, share resources, phenotype lines across all of their countries in West and East Africa, and use that to gain insights. And what's been really, really fun to see is that the initial round of sharing germplasm has led to dozens of variety releases in different countries where they wouldn't have had those lines previously. On the right, I'm showing in Malawi this year groundnut rosette virus disease. Groundnut rosette virus disease is endemic to Africa. It's completely devastating, and this year it was the worst it's been in 20 years. So the left is what it looks like for a normal peanut. It's completely stunted. This is at the end of the year. You don't have any nuts on it. You have no yield. You have no way to do anything about it. It's a virus. You can't spray for anything. On the right is a line that has the resistance, but again, most of those resistant lines aren't good for all the countries that have this disease, right? So you have to do a lot of breeding and selection with that resistance to get it in there. So the virus resistance that we could only identify using the pangenome graph, that we can tag using long read sequencing, potentially, and there's some evidence for this, provides resistance to this virus in Africa. So already we take those data we have, we go back and we look at which lines have the resistance, have this trait, are resistant to this fungal disease and others, and have shared those lines with different countries in Africa for testing already this year. So the ability, we take this resource and we immediately share it so that it can make an impact there. And as well as sort of SMUT has moved to different countries in Central America and South America as well. And they're having lots of trouble providing a resistant variety. Well, we've already identified and developed resistant varieties that are high yielding, that have the chemistry that they want, and that have resistance to other diseases as well. So we can immediately share those. And so if you go back to this program where these kids in their high schools are learning about how molecular biology can make an impact in their community, those plants, some of them that they named and they nurtured and they took care of and then selected, also have the ability to make an impact across the globe. And so we are very, very bullish on applying long-read low-pass sequencing across all sorts of different applications. Of course, for me, breeding and that my personal program, but plants and animals and others. And we're just so grateful for the technology. And thank you for inviting me. Thank you very much.